So is there anyone willing to volunteer to pray, to lead us in a word of prayer before we begin? Okay, Dingen, please lead us in a short prayer.
In the name of the Father, and of the Son, and of the Holy Spirit. Father, we come to you. We thank you for the gift of life. Thank you for the gift of this lesson. We pray that everything that shall be taught unto us shall come and manifest in our lives, and we shall learn and benefit from this. Through Christ Jesus Christ, our Lord, we have prayed.
Amen. Thank you, Dingen. I hope I've said the name right. So I'd like to welcome you, those who have joined us. And I would like to put down some ground rules so that we can have a smooth and fruitful discussion. Please do not put on your videos. It will consume your data and distract others from the proceedings of this discussion. And if you want to say something, kindly raise your hand and then you'll be called upon to contribute to the discussion. Otherwise, if there's nothing to say, kindly mute your mics so that we don't have interruptions. So I would like to bring your attention to the small quotation I have put in these slides. Success is not an event, but a sum of small efforts repeated day in and day out. So if we are going to be successful, we need to get to practice habits that will eventually make us successful. Success doesn't happen abruptly, but it comes from the repeated things that you do day in, day out, because we are products of what we have been repeating day in, day out. So this is week 2 of our discussion, and I have picked on other topics that I feel that we all should have covered by this time. I picked on the topic of ecology and the topic of coordination. So that's what we are going to discuss today. And if time allows, then we can talk about some other things. So here are the questions. So we have these structured questions. Question 1 says, explain what happens when a mammalian eye focuses on a far object in bright light. And then question 2 says, explain what happens when light shines on coreoptile, and a coreoptile, that is a shoot of a maize seedling from one side. Then lastly, question C requires us to talk about how these two responses differ. So we are going to start with the first question, the one that talks about the mammalian eye, how it changes when it focuses on a far object in bright light. So as usual, these are discussions. I expect participation from everyone so that we can be able to benefit. Then after that, we are able to share what the expected response should have been like. So who wants to tackle this question? Oh, Dingen, that's wonderful. Please share with us your thoughts about that question. A?
Yes, sir. According to me, in bright light, the circular muscles will contract and then the radial muscles will relax, causing the iris to become smaller and letting in less light, while the light from the far object comes in parallel to the eye, so it will cause the ciliary muscles to relax, causing the lens to thicken and convert the images much more.
Okay, thank you, Dingen, for your contribution. Others can also participate in the chat since all of us cannot be able to talk at the same time. Do we have any objections to what he has shared with us? He has talked about what happens in bright light, and he has also talked about what happens when the eye focuses on a far object. Do we agree with his submissions? I hope he didn't talk too fast that some of us missed out. Can we critique what Dingen has shared with us? Okay, I'm going to ask Dingen to repeat the answer. It seems some people are not attentive. By this time, you talk a little slowly so that people can pick what you're talking about clearly.
Yeah, according to me, first when you talk about the bright light, the circular muscles will contract while the radial muscles will relax, causing the iris to become narrow, letting in less light, while the light from the far object will come parallel to the eye, causing the ciliary muscles to relax a bit, causing the lens to become thinner and longer, hence less convergence of the image, forming the image on the retina.
Okay, thank you. I think this time it was much clearer. Do you have any objections? I think silence means that people agree with you. Yeah, so like Dingen has tackled the question, there are two angles to this question. There's where we look at light coming from a far object, but also we are seeing that this object is in bright light. So there are two angles to the question. So in bright light, like he has said, the circular muscles of the iris, what I noticed that he did not say that the circular muscles of the iris, he just said the circular muscles. So you must tell us where these circular muscles are. Yes, they are in the iris. So they are going to contract while the radial muscles relax. So that one, he talked about it very clearly. So what is the outcome of that? The pupil size will reduce, or the pupil aperture will reduce. I think you didn't talk about that, but you told us that this is to reduce, or rather this is to limit the amount of light entering the eye, which is very correct. And this is so as to protect the eye, the retina particularly, from excessive light. Because if there's excessive light, the retina will be damaged. So on that account, I think he has done very well. He just missed out just very small details. Then in order to focus on the far object, we see that the ciliary muscles surrounding the lens. You need to tell us where the ciliary muscles are. They are attached on the lens. They are surrounding the lens. They are going to relax, which he mentioned correctly. Whereas the suspensory ligaments are going to become taut, or they are going to become stretched. So these two events are going to cause the lens to flatten or to become thin. And I think he has talked about that very well. And this increases the focal length, okay, allowing the eye to focus on the far object. Allowing the eye to focus on the far object. Yes, Nelima Tracy.
Teacher?
Yes,
Teacher,
please.
I have a question. When you're answering a question of that sort, is it wiser for you to paragraph or is it wiser for you to bullet them? Because earlier, teacher had talked about paragraphing being a word in which you can lose marks because what if you like miss one step when you're like writing their stuff? So is it wiser to paragraph or to bullet?
I hope you did not misunderstand what the teacher was telling you. I don't know who told you, but I want to believe that you have misquoted the teacher. Now, when you are answering the question, for me, what my take is this. It depends on whether it is section B or section C. Now, these questions, if they are in section B, you have a limited amount of space to write your answer. You may have 3 or 4 lines provided. So if that is the case, then it means you don't have a choice. Cannot paragraph. You would simply write the answer and you work with your punctuations properly. So what will be like the first paragraph, wherever it ends, you put a full stop. Or then you start the next sentence or next paragraph from where you have stopped. But if it is section C, then paragraphs are good for your points to be very easy to spot out. Now, if you remember from English, a paragraph is something that talks about a certain related ideas. Okay? So if the ideas you're talking about or the point is being explained, then it should be explained within one paragraph. Or if it is about events that are happening in a step-by-step process, then it is better you write each step as a paragraph of its own. And then you arrange the steps in a logical order so that if the first step was written, then the following step should constitute a second paragraph. And of course, as you are explaining the steps, is very important. You mention the step and you talk about why it happens, how it happens, and probably why it happens. So I don't know if my submission clarifies things for you, Tracy.
Yes, thank you, teacher.
You're welcome. Yes, so Dingen, thank you for your submission. It has kicked off our discussion and I'm pleased with how you have tackled that question. So let's turn our attention to another question that follows that one. So the second question is to explain what would happen when light shines on a coreoptile shoot of a maize seedling from one side. So what would happen if light were to shine? Yes, Elizabeth.
Yeah, the shoot of the maize seedling.
Please raise your voice. We are not hearing you clearly.
Okay, I was saying like the shoot of the maize seedling would bend towards light because it's receiving light from a single direction, and also the auxins would move away from light to the dark side, whereby the dark side has a high concentration of auxins and therefore grows faster and longer than the illuminated side.
Okay, thank you, Elizabeth. Does anyone think Elizabeth has left out anything? And so that you may add something. Yes, Tim. Yes, Tim? Oh, Tim, okay.
Well, something on oxygens. So when light to a darker side, so the other darker side grows faster, bending towards the light.
Okay, thank you, Timothy. So I think we all agree that Elizabeth has done a good job. So this is what I had. So upon exposure to unilateral light is light coming from one direction, or you can call it unidirectional light. The oxygens diffuse to the dark side, or the side which would call the shaded side where there's no light, or we can call it the less illuminated side of the coreoptile. Okay, so you start by telling us what happens. So the higher concentration of oxygens on the shaded side will cause rapid cell elongation on the shaded side compared to the illuminated side. And I'm happy that as you are both explaining, you compared the side which has light and the side which doesn't receive light. So you start by telling us which side has more oxygens and which side has less oxygens. Then after that, you tell us which side has faster elongation or more rapid elongation, which in this case is the shaded side. And then you tell us what is the consequence. So if the shaded side has more rapid elongation of the cells there than the lit side, what does this mean? So this means that the side which has been shaded will grow faster than the side which has light rather. And so it causes the coreoptile to bend in the direction of light. I hope that is clear. So I thought I needed to emphasize some things. Yes, Benon, that is correct. So we can move on to the last part of the first question. The last part of the first question wants us to talk about the differences between these two responses. So first of all, what are these two responses? What are these two responses? And then from there, we want to talk about the differences in these two responses. Can someone tell us what these two responses are? Adrian?
I think one is a taxis response and the other is a tactic response.
Do we agree with Adrian that one is tactic and the other one is taxis? In any case, which one is which?
According to me, I believe the other one is a tropic response and one is a tactic response.
Please let us be more specific. When you say the other one, then this one, who is the other one and who is this one? Can we be more specific so that we can follow well? So far we have two contradicting answers, but I'm not taking any of them yet because they are not specific to which of the two responses is which.
The coleoptile response, the tropic response, while the I response is the tactic response.
Okay, thank you, Dingen. Do we agree that the coleoptile response is tropic, but that of the eye is tactic? Do we agree to that, the way he has put it? Please do not shy away. All answers are welcome. It is from making mistakes that we all learn. The worst mistake is fearing to make a mistake. Okay, maybe to be able to put their answers in perspective, what is a tropic response? How do we define a tropic response? I'm seeing the
Yes,
same
sir.
people all the time. Can we have more people participating? Okay, Adrian, Dingen, please, you raise your hand, okay? Let's not just talk without raising hands. Adrian?
Tropic response is a response to a unidirectional stimuli.
Okay, thank you, Adrian. I think you have left out something. Can Tracy, since your hand was up, can you supplement on what Adrian was saying?
Okay.
Tracy?
Tropism is a growth movement of part of the plant in response to external unidirectional stimuli.
Okay, thank you, Tracy. Because she has added in that it occurs in plants. So, Adrian, you had left us guessing whether it occurs in plants or in animals or in both. So it occurs in plants, and it's true that it's a growth response. And what both of you agree, which is correct, is that they are responding to the unidirectional external stimuli. Now, having understood what tropic response is, that means a coleoptile in this case is a tropic response. What about, I had words like tactic? What's a tactic response? What's a tactic response? Yes, Ivan.
Response is the growth movement of a plant part towards unidirectional stimuli.
Thank you for your submission. But tactic and taxis are the same, and tactic responses do not occur in plants. Okay? Dingen?
According to me, it's a response of a body part, like involves movement of the whole body part in response to stimuli.
Thank you, Dingen. Nsimenta, please give us your contribution.
Is the movement of an entire cell or organism in response to a directional stimuli.
Thank you, Nsimenta. So tactic or taxis involve the movement of the whole organism. It can be a cell or it can be multicellular, but the whole organism is responding to unidirectional light, and it occurs mostly in animals and in protists. For example, we can see maggots responding negatively to light. Maggots do not respond positively to light. So we call that negative phototaxis. Okay? Or it's negatively phototactic. So when we look at this question, here we are trying to compare the tropisms and nervous responses. So when we see the response of the eye to light, that is a nervous response. So we are going to compare nerve responses or nervous responses with tropic responses. Okay, so can we talk about the differences between nerve responses or nervous responses and those of the tropic responses in terms of their characteristics? Yes, Dingen.
The tropic response involves growth of plant parts, while the tactic response involves movement. Then you also talk about that and involves auxins, while this one involves impulses. Yeah, that's my contribution.
Okay, thank you. But we have agreed that the first one is not a tactic response, okay? So if you answer the question and you put heading tactic, then you put tropic response, you would not get the marks because of that simple mistake, okay? So you'd rather say response of the mammalian eye to light in one column and then response of plant coleoptile to light if you are not sure of the type of response. If you did that and you wrote the other answers, then you'd get the marks correct. You'd get your marks. Hope that is understood. So where you're not sure, do not commit yourself, because if you commit yourself, then you will be marked because you have committed yourself to the type of response. Can we have other answers apart from what Dingen has given us? He has told us growth, plant in the coleoptile there's growth, in the eye there's no growth, and he's told us in the eye there are, in the coleoptile he said there's use of auxins, whereas in the eye there's use of what? Of nerve impulses. Can we think of other differences other than those two? Yes, Ivan.
Can you please briefly explain to me what are nastic responses?
Or nastic responses are responses we see in plants, okay? But they are responding to diffuse light. Diffuse light is light that doesn't come from one direction. It's light that is all around. And when we talk about nastic responses, we can think about the Mimosa pudica plant. The Mimosa pudica plant responds to the touch. It's called haptonasty or thigmonasty. I hope that is clear. Then we can also have the response to light by flowers, like the morning glory flower. It opens its flowers in bright light early in the morning and then closes its flowers at night. And the idea here is it is opening its flowers so that the pollinators can pollinate it. At night, those pollinators will have gone to sleep. So there's no need to open the flowers when there's no pollinator to pollinate it. Lindsay is saying that in the plant, the response is irreversible, while in the mammalian eye, it is reversible. That is correct, okay? And in plants, the response is slow, while in the eye, it is fast. The spelling of fast, Lindsay, is supposed to be F-A-S-T. So if you write this other spelling, the meaning will change. It will mean another thing. So F-A-S-T is what you should have said rather than F-I-R-S-T, because the meaning here now completely changes. Then Gilbo is telling us that in the eye, the response is fast. Yes, that is the spelling I wanted. While in the shoot, the response is slow. Thank you for your contributions in the chat. So if there are no other contributions, then we can see what I had in mind. So this is what I have. So we can mark ourselves. The first response I gave here has been captured. In the second, it hasn't been mentioned. So nervous response involve nerve cells, or what we call neurons. But in the tropism, we don't see neurons. Nerve responses are not controlled by hormones, whereas tropisms are controlled by plant hormones, which we call the auxins, or what we call phytohormones. Then the nerve response involves transmission of electrical impulses, but in tropisms, it doesn't involve electrical impulses. Then the second last was mentioned. Then the last one was not mentioned, but was implied. So these are some of the answers you ought to have considered to answer that question. Okay. Are there any questions this far? Yes, Tracy.
Teacher, could you talk about the existence of receptor organs in nervous responses and no receptor organs, I think?
Okay, you can talk about that as well. But also what I want to caution you, when you're answering these questions, they always put marks allocations. And if they put 4 marks, it means that we are going to consider the best 4 answers that are written first. So you should always write your best responses first. Okay? And what you're not sure about, you write it as your last response. And it's better you also write 4 answers rather than writing more than what is required. Because we are training you to be people who can follow instructions so that you don't cause accidents or cause problems for yourself. Okay, so we are going to go to the next question if there are no questions in as far as we have moved. So we have another question here. This question has a diagram on it. So the diagram below shows the response of a germinating seed which was placed horizontally on the ground. So we are required to state the type of tropism that caused the kind of response that is shown in the diagram. Then after that, we are expected to explain why the seedling behaves the way it has behaved. So let's go to the chart and see. Tracy says geotropism. Gilbert also says geotropism. Oh, someone was asking about the previous question. Is it okay to draw a table in the paper? Table of differences. If the space allows, yes, it's okay, especially in section C, it is allowed. But in section A, section B rather, you may not have that kind of space. So if you don't have that kind of space, then it's better you write in long form. You say that, and it's always important that you punctuate your differences with words like whereas or while, okay? You don't just write sentences and leave them hanging. The beauty about tables is that they make the work very organized and easy to read, and they are more easier to score. So so many of us, I can see Rami, Peter Chiveru, agreeing with the earlier submissions. Elizabeth and Cementa, Adrian. So the answer is geotropism. So you're all correct. Okay. So it is showing us geotropism. But now the question is, geotropism where? Because we are having the shoot and the root. Yes, can be geotropism, but what would you say? Which one is responding positively and which one is responding negatively to geotropism? Okay. And for this thing to make even sense is if the question says that this was put in a dark room, okay? If they said it were put in a dark room, then answers like phototropism will not apply. But if they leave it open, then it means even phototropism can be argued, okay? It can be argued, but the answer really I wanted here was geotropism. I think I forgot to put that this thing was growing in a dark room, so that we have only one answer. Okay. I can see that chat is quite busy. I'm happy with this. So Elizabeth says that the root is showing positive. We should add geotropism. And then Adrian says the root is responding positively. I'm assuming he wants to say geotropism, but in exams, you never leave answers which are not clear. So write what you want to say, okay? Write what you mean, okay? Don't leave us guessing. Then Becky says the root due to gravity, that is positive response. And Tim says the shoot is negatively. So let's write complete answers. I can see the idea is there. Okay, so can we explain? Okay, can we explain why we say that the shoot shows negative geotropism while the root shows positive geotropism? How is that possible? Yes, Elizabeth.
Yeah, in the shoot, cell division takes place. Sorry, in the shoot, cell division takes place and elongation takes place more on the lower side than the upper side, which makes the shoot bend upwards away from gravitational pull, which makes it a negative geotropism. And then in the root, the higher concentration of auxins in the lower side reduces the rate of cell division and elongation.
Thank you, Elizabeth. Did you say cell division or cell elongation?
I said it reduces the rate of cell division and elongation.
Okay, thank you for your submission. First things first, auxins are important for cell elongation, not cell division. Cell division is brought about by cytokinins, okay? Cytokinins, not auxins. So do we have someone who wants to add on to what Elizabeth has mentioned? This time people are shy. I don't know why. Okay, let's look at the chat. I'm seeing only one chat. You can see someone is telling us that auxins are attracted to the side closer to gravity, thus causes rapid growth of the side with less auxins in the root, while the shoot is the opposite. Okay, Dingen, that is correct, except I would expect you to write and tell us what happens in the shoot rather than telling us it is the opposite. Okay, in an exam, let us not write short answers because they will also give us short marks. Because when they are scoring, they will score for those words you have avoided to talk about. So you'll get less marks. So it is true, auxins will say what we call sedimentation. They will settle on the load of the plant that is closer to the pull of gravity, okay? And when that happens, it will have different effects, like Dingen has said. In the shoot, it will cause that part to undergo rapid cell elongation compared to the upper part where there are less auxins. And so that part will grow upwards away from the pull of gravity, tropism. Whereas on the lower part, we can inhibit cell elongation. So that part will elongate slower than the upper part where we have a low oxygen concentration. And so that will cause those parts of the root to elongate faster than the lower parts, causing it to bend towards the gravitational pull. So basically, that is what the answer ought to have been like. I've put it on display. We can read the answer. You could also take some screenshots so that you can look at it. So basically that is it. Okay, are there any questions this far? Okay, silence means there are none. So I think we can move on to the next thing. So we are going to describe how the ear perceives sound. This is still part of coordination. Remember, you are expected to be able to answer how the sense organs work. The sense organ that we are expected to know is the eye, the ear. Those ones are very, very important. Of course, even the skin, because we know that skin has a number of sensory cells. We have the sense of touch, the sense of pain. We have the one which responds to heat. So you should be able to explain how they also work. Of course, even the tongue, we know that it is a sense of taste. So you should be able to explain that as well. But for now, we want to explain how the ear perceives sound. Who wants to explain to us how the ear perceives the stimulus of sound? This is a typical question that can even carry 15 marks in section C. So when a question carries 15 marks, it means that you need to write all the required steps. And this is a type of question which is a process question. So it means you must write the steps in their correct order. Yes, Dingen?
So according to I understand the question, it's like, of course, sound from a distant object, which is the pinna, and this direct sounds to the auditory canal. And then the sound waves reach the ossicles and they cause this to vibrate, and this eventually end up also hitting the eardrum, and then the eardrum sends vibrations to the cochlea, and then from the cochlea they go to the auditory canal, to the eustachian tube, and then impulses are generated and then sent to the brain for interpretation.
Okay, thank you, Dingen. I can see from the hands that have shot up as you were presenting, there are some things they did not agree with. Let's hear from Gilbo, then Lindsay, then Elizabeth, in that order, in case there are things to add on or subtract. Gilbo?
Yeah, sound waves are received and...
Gilbo, can you raise your voice? We can't hear you well.
Sound waves are received and concentrated by the pinna through the auditory canal to the eardrum, where they are caused to vibrate. The vibrations are kept by the ossicles to the oval window. The ossicles amplify these vibrations, and the vibrations are transmitted by the sensory cells which produce impulses that are transmitted by the auditory nerves to the brain. The brain interprets these impulses, and they can determine and this determines how low or high the sound is.
Okay, thank you, Gilbo. Lindsay, do you have something more to add or subtract?
Yes, teacher.
Yes,
So
please.
the sound waves are collected by the pinna and directed through the auditory canal to the eardrum. The eardrum vibrates and transmits the vibrations to the ossicles, and the ossicles also vibrate, transmitting the sound to the oval window. The oval window vibrates, causing the liquid in the cochlea to vibrate, transmitting the vibrations to the organ of Corti, which is found in the cochlea. So this organ causes transduction of the sound waves into impulses, which are transmitted through the auditory nerve to the brain.
Okay, thank you, Lindsay. Okay, to a large extent, you and Gilbo have said the same things. There's something that I think both of you have missed out, which I want to bring to your attention. That is the role of the ossicles. The ossicles don't just merely vibrate, the ossicles amplify the sound waves. So they are in form of a lever system, although you don't need to add that. They cause the sound, which is otherwise low, to become amplified. Okay, so I think we missed out that. So what is important for this question is to know the steps in their correct order and the role of the different parts of the ear. So the pinna is to collect and concentrate sound waves, okay, from the environment. If you want to appreciate the role of the pinna, I want you to get your hand and put it on your ear, okay? Put it on your ear, and so that you imagine that your hand is now an extension of your pinna. So put it there as you are listening to what is around you, then remove it. If you do that, you will notice that when your pinna, your hand is on your pinna, you are able to hear a lot more clearly than when your hand has been removed. So it means that the larger the pinna, the better the organism is at collecting sound waves. That's why you find rabbits have longer pinna, so that they can even detect the slightest of sound in their environment, so that they can increase their chances of escaping from predators, because these predators tend to walk very stealthily or walk very softly on the ground so that they don't alert the prey. So, and the prey also have to find a way of detecting the slightest sound so that they can increase their chances of escaping. Yeah, so when the sound reaches in the auditory canal, then of course it's picked by the eardrum. I think Dinden did not tell us the eardrum at the right time, okay? So the eardrum will vibrate. So you tell us the organ and what happens to it. So when it vibrates, of course, the vibrations of the eardrum are amplified and by the ossicles. Because this is taking 15 marks, you have to tell us which ossicles they are in their correct order, starting from the malleus, incus, and finally the steps. Or you can call it the anvil, rather the hammer, anvil, and stirrup. Remember, it is M-I-S, miss, or has. So if you use malleus, the next word should be incus, and the final word should be steps. If you use hammer, the next word should be anvil, and the last word should be stirrup. So pick what is easier for you to remember. Of course, the vibrations are picked by the oval window, which leads to the inner ear. Now, of course, there are two windows in the ear, the oval window and the round window. The question is, how do you know which window to write? Remember your alphabet, O comes before R. So from the oval window, from the ossicles, you go to the oval window. Now the round window is usually there to deal with echoes, because when sound enters when those vibrations enter the ear, there must be a way of dealing with the other form of energy, and that comes out through the round window, and it builds up pressure in the middle ear, which pressure must be equalized by the eustachian canal. But of course, for this question, you don't have to write all that. These are things I'm telling you so that you can have a proper appreciation of how your ear works. Peter Chiveru is asking, how about an elephant? Does it mean it has highest ear sensitivity? The elephants, yes, they are quite sensitive, but you see, the elephant ears also serve another purpose. Who can tell us, apart from detecting sound waves in the environment, what other secondary purpose does the elephant ear serve? Okay, can see someone. Oh, King of Spades, what a name. Cool the body, that is correct. So they will fan their ears and generate convection current, okay, around their body and cool the body. Remember that an elephant has a small surface area to volume ratio, so it easily generates a lot of heat in its body, and this heat needs to be dissipated, or the heat must be shed off. So one of the ways to shed off the heat is by fanning the ears. But sometimes the elephant can also roll in mud or go in a water body and take a swim, if you may. And by so doing, the water will carry away some of the heat from the body. So you can see that ears serve more than one function. Although the primary function of the ears is to receive sound waves, but they can also do other things other than that. Okay, so let's move on. So you notice I have numbered these things so that they can guide you. So the vibrations of the oval window makes the fluid in the inner ear. I think one of us talked about that in the chat. The inner ear has the fluid, and that fluid, when it receives the vibrations, it is going to cause hairs in the cochlea to be disturbed. So when the hairs are disturbed, then that is going to cause the transduction that one of you talked about, okay? So that transduction is changing energy from one form to another. Remember at this point, the energy is in form of vibrations, and now it's going to be converted into electrical energy, which can now be conducted by the auditory nerve. So the organ of Corti, which is part of the cochlea, it transduces or converts energy from sound into electrical energy, or what we call the impulses. And once, of course, it goes via the auditory nerve, then the brain, there's a part of the brain which deals with sound. It's the one which is going to receive the sound and interpret it. And then you'll be able to interpret that sound, whether it is a whisper, or it is some loud noise, or it is a barking dog, and then the brain, of course, will help you to know how to deal with the information, whether you should run away or you should turn and see what has caused that noise. So basically, that is how you should have answered the question. So there are about 5 steps, and at each step, we need to know which part of the ear is involved and what next part of the ear is involved and what it exactly does. So do we have questions on this before we proceed? Those who are taking screenshots can make screenshots before I go to the next page. Okay, it seems everyone is silent. And silence probably means that we have understood. Okay, we have another question here. I think this is a very familiar question. Okay, someone has posted something in the chat. Oh, it's just an affirmation. Someone is asking to explain the tympanic canal. Okay. Well, at our level, I don't want to bother you with content that is not relevant for your level. Okay. But otherwise, the tympanic canal is part of the cochlea. Okay. And there's a tympanic membrane, but the details of that information are not relevant for O level. That information is required at A level. So let us concentrate on what will earn us marks. So here we are being asked to describe the series of events that happen when you touch a sharp pin. Okay. Of course, this image should not have been there, but I posted it so that it can give us some guidance. So, Adrian, can you describe the series of events that happen when you touch a sharp pin?
When you touch a sharp pin, the receptor cells will be stimulated to send impulses through the sensory neuron.
Let me interrupt you briefly, Adrian. Where are these receptor cells? Please answer that and continue with your presentation. Oh, okay, Gilbo. I think we've lost Adrian.
The receptors are found on the skin.
Please raise your voice.
They are found on the skin.
They are found on the skin. Yeah. Okay, that's correct. So do you want to carry on with the presentation?
Yes.
Okay, please do. Please go on. Okay, I can see something in the chat. Okay, skin under the skin. That's correct. Okay, who wants to carry on from where Adrian and Gilbo have stopped? Oh, Gilbo wants to? Okay, please carry on.
From there, they are transmitted by the emotion.
Kindly raise your voice because we want to hear what you're telling us.
Yeah, okay, let me start over. When one touches a sharp object, the stimulus is detected by the pressuring receptors of the skin, which send impulses through the sensory neuron to the spinal cord. From there, they are transmitted by the motor neuron to the muscles of the arm that will be withdrawn from the sharp object.
It's unfortunate that your good answer has not been well heard by many people. So I'm going to ask someone who is louder to present. Adrian?
The sensory neurons receive the impulse when someone touches a sharp pin. They are sent through the sensory neuron to the synapse, where they are transmitted to the motor neuron by the relay neuron, which motor neuron transmits the impulse to the effectors that produce an impulse, resulting into one removing their hand from the pin immediately.
Okay, thank you. I think Dingen has something to ask to add on.
And I just wanted to add on towards his end. It's like once the impulses reach through the motor neuron, reach the muscle, they cause rapid contraction of the muscles, causing one to draw back the hand rapidly.
Okay, that's a correct submission. Maybe what I want to draw to your attention is something that probably you said without noticing when you say that the pin, when the pin touches the skin, then the sensory neuron receives impulses. We need to appreciate that when we are discussing questions on nerve impulse transmission, we must first talk about the impulse, the stimulus, then receptor organ, then sensory neuron, then relay neuron, then motor neuron, then effector organ, in that order. Okay, so first is the stimulus. What is the stimulus in this case? The stimulus is that of pain. Okay, when the pin pierces the skin, it will stimulate the pain receptors. There are pain receptors in our skin, okay, because of a sharp pin. Then the pain receptors are going to fire, okay, an impulse, okay, along the sensory neuron, okay? And remember, the sensory neuron transmits this impulse to the spinal cord, okay? But it passes through the dorsal root of the spinal cord, okay? And in the spinal cord, it is received by the relay neuron, or you can call it the interneuron, okay? By the relay neuron or the interneuron, which sends the impulse to the motor neuron, which passes the impulse to the effector organ, in this case being the muscles, and that causes them to contract, okay? You see the 4 steps? So when your hand touches a sharp pin, they are what we call sensory receptors, okay? In the skin, they generate nerve impulses that move along the sensory fiber or the sensory neuron to the dorsal root ganglia towards the spinal cord. Notice the spelling of cord, okay? And then the sensory neurons pass signals or impulses onto the interneurons or relay neurons. Choose one word. And then these neurons are going to pass them to the motor neuron, and which will also pass them over to the effector. So I think you see what the layout is like. I saw a hand up at some point. Yes, Ivan.
Can I talk about the gray matter?
It's not relevant. It's true there is gray matter in the spinal cord, but how does this gray matter add to the events? I hope you get what I'm saying. The gray matter is the part of the spinal cord where we find cell bodies. That's why it's actually gray in color, okay? But mentioning it or not mentioning it doesn't take away marks or add you marks. However, if you mention it and you mention it wrongly, it may cost you marks instead. So let us keep it very, very simple. Do we have other submissions? Okay. If there are none, then we can carry on to the next question. So now we are switching to ecology. We have a question here. Oh, Dingen has a hand.
Sir,
Intermediate neuron is the same, it's the same as the interneuron or relay neuron. Let me just throw some more light on the interneuron or the relay neuron. I am sure, or at least I hope, that in your different schools, you carry out sporting events. And one of the sporting events is the race called the relays, okay? In the relays, we have different competitors running and exchanging the baton. There's that small stick which is called the baton. So if you have, for example, 100 by 4 relays, it means that you're going to have 4 people running the race, and each one runs 100 meters. When they finish the 100 meters, then they pass on the baton to the next competitor until the final 100 meters. So the passing on of the baton from one athlete to the other is what we call relay, to relay. So that neuron is between 2 neurons. It's between the sensory neuron and the motor neuron. So that's why we call it the interneuron or intermediate neuron. But we also call it the relay neuron because it picks impulses from the sensory neuron and then passes on these impulses to the motor neuron. So by so doing, it is relaying impulses. So that's why we call it the relay neuron, or that's why we call it the interneuron, or that's why we call it the intermediate neuron. Inter means in between. Intermediate also means in between. So that's why those names come about. I encourage us not to cram, but to make sure that we understand these things from the way they are named. Sensory neuron is attached on a sensory organ, and sensory organs are able to detect the stimulus. So they are sensing. Then we have a motor neuron. The motor neuron, for it attaches on the effector organ. So it causes some form of motion. Maybe that's why we call it the motor neuron. So are there any other questions in as far as this is concerned? Okay, Dingen, your hand was up before I answered this. Yes?
So I wanted a clarification about transmission of impulses across the synapse, like what's involved, the whole process about it.
Okay, when impulses are being transmitted, they are transmitted or they are conducted along nerves. However, the nerves are not in physical contact with each other. These nerves are separated from each other by very narrow gaps, what we call the synapse. So it means that since the impulse is electrical, electricity cannot be transmitted across space, okay? So the impulse instead is converted into, it causes, okay, what we call chemical conduction. So between the one neuron and the other, we have a synapse, and there we have what we call a neurotransmitter substance. So the neurotransmitter substance is a chemical which moves from one neuron to the next neuron. And so the conduction takes place. So we have neurotransmitters like acetylcholine. So this acetylcholine is one which causes transmission between impulses across the synapse. And actually the medicines we take sometimes cause these impulses not to be conducted, or more of them to be conducted, so that now we either feel pain or don't feel that pain. Okay, but that detail we shall find it when we do biology at A level. Someone from Viva College is asking, is the motor neuron also called long arcs of motor or not? I'm not sure I understand what your question is, but I would suggest we stick to simpler descriptions, okay? So we have a sensory neuron, which in some books is called the afferent neuron, and then the motor neuron, which is also called the efferent neuron. But I would suggest we stick to these simpler words. When they set questions, they also try to keep the language simple, because the intention is not to fail you. The intention is actually to see whether you have grasped the concepts, okay? Whether you have understood the concepts, not to see how many of you can fail. So let's stick to the simpler words that are in the textbooks that we have available to us. So I think we should close the chapter of coordination. Now we need to move on to the chapter on ecology. So I want us to look at this information on this slide critically, and we are going to use it to answer the next question. Nsubuga, what is the issue?
I have a question about coordination.
Yes, please.
I have a question about coordination.
Yes, please.
Describe the process leading to the formation of a zygote in a bean plant.
I'm sorry, can you repeat your question?
Describe the process leading to the formation of a zygote in a bean plant.
Zygote in a bean plant.
Yes, the process.
Well, that is not coordination. That question is not from coordination. If I've understood your question, that question is probably from growth and development, okay? Or some aspects of reproduction. So because we are not dealing with that, I would suggest we can keep that until later. Yeah, but very briefly, if I've understood your question, probably it's trying to talk about the events that follow from the time of fertilization, where we have the first and the second male nuclei fusing with the ovum and the polar nuclei in the embryo sac. So, but since this is not the point of discussion, I would suggest that we leave that for a later time. Okay?
Okay, sir.
Yeah. Seeing someone in the chat. A wrestler faces a very aggressive opponent in a fight. Wow, that's interesting. Explain the physiological changes to prepare him for the fight. Okay, it's a good question. I'll take it and bounce it back to the rest of us so that we can do it justice. So imagine you are the wrestler and you have been put in the ring and you're supposed to face the champion, okay? Someone who is a champion. So can we explain the physiological changes, they underline the word physiological, that prepare you for the fight? Remember, you cannot escape from that fight, okay? Maybe it can allow you to hang in the towel, to throw in the towel and say, I give up, but let's assume that is not even allowed. So how would this person body prepare him for the fight? Who wants to answer the question? So what does this question require us to talk about? I think it's sort of related to what we discussed last week. Can we give it a try? Yeah, no need to be cowards. So I don't know why we are shying away from answering the question. Yes, Suga.
Master, I'm trying.
That's great.
The inner zone of medulla, the adrenal gland, is stimulated by the nervous system and produces adrenaline. When the sense organ of the person transmits to the brain, impulses which are associated with the danger or other situations needing vigorous action, motor impulses are relayed to the adrenal medulla, which releases adrenaline into the blood. When this reaches the heart, it quickens the heartbeat. In other regions, it diverts blood from the alimentary canal and the skin to the muscle. It makes the pupils dilate and speeds up the rate of breathing and oxidation of carbohydrates. That's my trial.
Okay, thank you. At least you've given us a good start. Can someone add on what he might have left out? Can someone add on? To a very large extent, he was on the right track. So this question is basically about the effects of adrenaline. So when this person sees this aggressive opponent, the adrenal medulla is going to secrete adrenaline to the bloodstream, like he has mentioned. Then adrenaline will have a number of effects on the body. Then you mention, like he has mentioned. So of course, diversion of blood supply from the stomach and move it into the limbs. And so like he has explained it, okay? So we are going to see the liver breaking down glycogen to glucose, and the glucose being broken down to generate energy. We are going to hear that, we are going to see the heart beating faster. I think you missed out that. In order to pump more blood to the limbs. Because remember, in wrestling, you use the hands, you use the feet. We are going to see increased breathing rate to generate a lot of, to take in more oxygen, okay, and carry out more carbon dioxide. So I think Nsovga has done the question well. So I think let's move on to the next chapter. That is the chapter of ecology. So we have an investigation about a rat population under laboratory conditions. So in your schools, you may be growing some, you may be rearing some rats that are used by the A-level students. Okay, so imagine you have such a situation, and this is done over a period of 6 months. And assuming we also have another situation where in a nearby bush, maybe, we have other rats. So we introduce 20 rats in our lab cages, and then we introduce 20 rats in the natural environment. Then we keep going and checking every after 1 month how many rats are there. Okay. So what this table shows us what was obtained at the end of the 6 months. Okay, at the end of the 6 months. You notice that the population growth is different, it's not the same. So you are required to plot the graph to show the population growth under the two conditions. Unfortunately, we don't have enough time for each one of us to do this work, but I would like to encourage you to take a screenshot and try to plot this information on a graph paper. Okay, for me, I have plotted, and I'm going to share with you what my graph looks like. Someone interrupting my call. I think we shall put them, do not disturb. I'm sorry, there was a call interruption, but I've dealt with that. I hope you can hear me now. Unfortunately, I am not able to do that on WhatsApp. I encourage you to take screenshots on your phone. Or if you're using, but this information is going to be available. You can talk to the people, and they will get you the recording because it's being recorded. Okay, I think the recordings are on YouTube, so you should be able to get them as well. But if you can't, then I encourage you to take screenshots. Okay, so what similarity is there in the population growth curves of the two populations of rats? So we'd have to look at where the rats have the same population growth. And then what differences are there? We shall have to talk about those, then explain why there is a difference. Then besides starting with the same number of organisms, or rather rats, what other two things must be done to obtain the right comparison? And then lastly, what method is suitable to estimate the population of rats in a natural environment? So these are the questions we are going to discuss for the remaining part of our interaction. Someone wonders whether they can submit the work to me later. Well, if you have access to me, it's okay, but you can share them with your teacher. Okay? Yeah. Okay, let's continue. So here I have posted the questions. So let's attempt these two questions, and then we proceed. So what similarity is there in the population growth curve of the two populations of rats? What similarity do we see in our graph? This is what the graph should look like. Are there some similarities? Yes, Adrian.
There is a gradual increase in the population of the rats for the first 3 months in both the natural and laboratory environment.
Okay, thank you, Adrian. You're saying in the first 3 months, there's a gradual increase. Do the rest of us agree with Adrian? Do we see that there's a gradual increase for both the lab and the natural population rats in the first 3 months? Okay, Becky agrees with you. The rest of us are quiet. Okay, I see someone giving a thumbs up. That is Peter Chiveru. Okay. So Guy Ivan seems to agree or wants to submit something. Team agrees. Benon says there's an increase. I don't think we can take that because throughout the graph we are seeing there's increase, but what kind of increase is it? So we must qualify the increase, whether it is rapid or gradual. Yes, Ivan.
I want to answer the first question.
Yes.
I think for me, the similarity is between 1 and 3 months. I think 1 and 3 months, because there is a gradual increase in the number of the rats of both the laboratory and the natural habitats.
Okay, so that means you agree with us from what you have said. And that is actually true, okay? So from the 0 to 3 months, both populations of the rats increased gradually. Now, what differences are there in the growth of the two populations? Do we notice any differences? If you notice them, what are those differences? Ivan?
For me, the difference which I see is the one for the population of the rats in the lab between 3 and 7 months. There was a rapid increase, while the one for the natural habitats between 3 and 7 months, there was a gradual increase.
Okay, thank you. Becky? Becky, can you share with us what you had in mind?
So it's the same answer that he has just given.
That is one difference. There are more than one difference. So can we find another difference? Is that the only difference there is? I can see another difference. So what happens after 5 months or after 6 months? Okay, Peter?
I'm not answering the question, but I'm asking a question.
Please speak loudly. I can't hear you.
Master, I'm saying that I'm not answering that question, but I'm asking question. Master, can we use, in giving the differences, can we use like include the predators?
No, we cannot include predators because the question is just talking about what we see. The predators are probably what we think causes what we are seeing altogether. So the question is not asking us what has caused the difference, but is asking us what is the difference. I hope that is clear. So when we look at the questions that follow that is where we might consider what has caused the difference. Yes, Ivan.
Okay. After 6 months, I see a constant in the number of in the population of the rats in the laboratory, while for the one I'm sorry. After 6 months, I see a population, the population of the rats in the natural habitat, it is constant, while the one for the population of the rats in the laboratory, for it still remains at a rapid increase.
Okay, thank you. Tim, you have something different?
What I have is after 5 months.
You tell us what you're saying.
Population of, yes, after 5 months, the population of rats in the lab rapidly, as in natural habitat, increase gradually. That's what I have.
Okay, thank you. So I believe we are all seeing those differences. So from 0 to 3 months, as all of us, from 3 months to 6 months, the rats in the natural habitat increase gradually, while those in the labs increase rapidly. I think people said that. Then from 6 to 7 months, we see the population of rats in natural habitats remain more or less constant. Why am I using the word more or less? Because on our graph, on the table, if you look at the table, we don't see the number remaining the same. There's a slight increase, but the increase is so small that we can say more or less constant. That's why we use the word more or less constant. While that in the labs continues to rise rapidly. Now, besides starting with the same number of rats, remember we started with 20 rats in both cases. What else should we do so that our results make sense? What else should we do so that our results give us the right comparison? Can we have some contributions? Peko? Peko has run away. Adrian?
Can we say putting the rats in the natural environment in an area that is more or less free from predators?
You see that question? Read that question again.
So it's the same number.
So whatever you do to one population must be done to the other population. All together. Jaden?
What if I say that
you have to be with the same kind of rats.
Can
like
you
of the same breed.
Can you elaborate a bit, because you seem to have the idea?
Like I'm saying, you have, if you're using like a local breed of rats, they have to be the same in the laboratory and in the nature, natural.
What you said is correct, but I want you to explain so that some people can appreciate your answer. Why do you want to use the same breed of rats?
Because
why aren't you using different breeds?
As we all know it, the fertility rate and the growth rate is different. So using different breeds of rats will cause an imbalance and incorrect results.
Okay, that's correct. I think you deserve flowers. The members who are in keeping quiet should all clap for you, because you made a very good contribution. What other thing can happen? What other thing should happen? Okay, BJ is saying same amount of food and water. The rat should be of the same age. That's also correct. You can also clap for BJ. Okay, same species. All right. And Subga has something to tell us.
For me, what I can say that for the case of the rats in the laboratory, they can be used. They can be used in experiments, so that we can create a balance in the population.
Well, that's now something different, but not related to this very question. Of course, in the labs, we always keep them there for us to use them for experiments. But here, this is a study trying to understand what if populations of rats were surviving in conditions similar to those in the lab, would they still grow at the same rate as those in the wild? So that's what this whole thing is about. Okay, so they should be of the same size, same age, same species, as some of you have correctly talked about. Now, what method is suitable to estimate the population of rats in a natural environment? Someone has said capture mark recapture method. That's correct. What's another name for that method? If you don't use that name, what else can we call it? What's the name of the other method, of that method called capture mark recapture method? Yes, Ivan?
Capture, mark, recapture.
Yes, apart from that name, what other name is it? Let me give a hint. It starts with letter L and ends with letter X. No one remembers. Okay, so it's called the Lincoln Index. I have put it in the chat. It's called the Lincoln Index. So that's the other name of that method. And then lastly, what assumptions are made when you're using that capture, mark, recapture method? What assumptions are made for that method to be accurate? Yes, Chiveru?
I think none of the rats died.
That none of the rats that okay, none of the rats died. What else? So between the first and second capture, I think that's what you should say. Between the first and the second capture, from the time when you first capture, the second time you go to capture, in between, no rats should have died. What else? So if you want the answer to be more accurate, you say between the first and the second recapture, none of the rats died. Echo?
Trapping.
Speak up.
Trapping.
Trapping? I'm not sure I've understood you. Do you have anything in the chat? The chat is quiet. So you have to also assume that no new organisms are born in between the first capture and the second recapture. Then thirdly, rather, you have to assume no organism, no rats migrated, okay? That they did not migrate, or no new rats came inside. You know, if other rats come from the neighboring community, they can cause you to have wrong results. And then lastly, the marks you put on the rats should not be too conspicuous to make them an easy target for predators, or they should not make them fail to mix freely with other rats. So that mark needs to be very small and not very easily noticed by other rats. Otherwise, they will refuse to mix with it. Okay, there's some part of the question that I think are not included. Maybe we can go back and try to understand why is there a difference in these two populations after 3 months? Can you explain the difference in these two populations after 3 months? Adrian?
The difference is caused due to presence of predators in the natural environment that constantly feed on the rats, therefore reducing their population, while in the laboratory there is protection from predators, hence leading to
Adrian, you had started well. Can you complete your thought?
I'm saying that in the natural environment, there are predators that constantly feed on the rats, hence leading to reduction of their population, while in the laboratory environment, the rats are protected from predators, which leads to an increment in the population of the rats in the laboratory.
Okay. So maybe what I can add on to your sentence is you say there is a faster increase in the rat population,
Yes.
in the lab. They are both increasing, but you say that is faster. So in the labs, yes, there are no predators. Is it only predators that cause the rat population to decrease in the natural environment? I see Lindsay saying the rats in the lab are well fed, unlike those in the natural environment.
Not really.
let her defend her point. I think there may be some sense in what she's saying. Lindsay, can you tell us more about your point? Who can build on Lindsay's point? Becky?
According to Lindsay's point, I think it's because in the natural, as for the in the natural habitats, there is a lot of competition for food, while in the laboratory they are provided according to their cages. There is no competition. At least it's a must that all of them will get, and I think
Okay, thank you, Becky. So in the natural habitat, there is competition. But if you leave it at that, it's not clear. You have to tell us there is interspecific competition. Interspecific competition between those rats and other animals that feed on the same food. Okay, someone is saying, what of diseases? Where would you expect more diseases? Then another is saying harsh conditions like drought and diseases. Well, all those can be explanations for why there is a decrease in the wild compared to the lab. Because in the lab, maybe they clean the environment, they feed them well, nice balanced diet, there are no predators, they are safe, they each have their own cages with enough space. So all those answers can apply, okay? Adrian, your hand has remained up. Someone
Sorry,
is saying, okay.
There's a problem. I had a problem.
Is it sorted? Okay. So here we are seeing Manova saying both interspecific and intraspecific competition in the natural environment. That is true. So in the lab, we have only intraspecific competition, but in the natural environment, both of them exist. Okay. So I think that is all I had prepared for you today. We shall call it a day. Maybe next week, we'll see if we can have another meeting, okay? All right, so can we have one of us, someone who has been very quiet, to at least pray for us? Because God has given us very good network today. Becky, you have not been quiet. Maybe you are typing.
it's okay, sir.
Yeah. But if others are shying away, then you can pray for us.
Let me give them a chance and see if none, then I'll come back.
Okay, Karuhanga Joel, please pray for us.
Let's humble ourselves for prayer. In the name of the Father, and the Son, and the Holy Spirit.
Karuhanga, are you praying? We can't hear you.
Let's humble ourselves for prayer.